Wait till it says.
All right.
Great. Go ahead.
All right. I'm going to get to introductions in just a second, but first want to acknowledge our funding sources. So this is a part of the Sea Grant project, a Woods Hole, a Hui Sea Grant project. Some earlier aspects of this was funded by the Ruth Turner Foundation. Ruth Turner was a pioneering malacologist, professor at Harvard, one of the first female tenured professors in that field at Harvard. And then there's funding from Hui and UMass Boston. I also want to, if I can click my click here. No, it doesn't do that. There we go. I also want to acknowledge the research partners. So Quite Bay, particularly Megan and Jen, have been awesome. We've got some undergraduate volunteers. And then I'll be talking about septic systems, and the Massachusetts Alternative Septic Test Center on the base is been a fabulous partner in this. So when EPA, the folks have departed from the EPA, but they work in the EPA. So first of all, who we are and where we're from. And I'm going to start off, and then my partners in this, my co-PI and the graduate student will introduce themselves. So I'm Jed Goldstone. I'm a research specialist at Hui. I'm also recently chair of the Family Board of Health, although I was chair for a number of years a while ago. So we were asked to sort of give our paths to this, give our paths through science. I'm actually a fourth generation academic biologist. My maternal great grandfather was a professor of biology. We recently discovered that he actually got engaged. No, he didn't get engaged in Woods Hole, but traveled to Woods Hole after he was recently engaged. It's a little clipping in the newspaper tucked in someone's book somewhere. The family lore goes that he was the first person to teach evolution west of the Mississippi in the 1880s. My grandfather was a professor of biology at Harvard and taught at MBL summers for 4 decades or so. And unfortunately, never purchased a house in Woods Hole. That would have been lovely, but he grew up in Maine, and he always looked to back up there, and he went back up and worked on the farm and then retired back up to Maine. My mother was a research technician at Stanford, Harvard, Bermuda Biological, and NIH. And on my father's side, my grandfather taught at Johns Hopkins. So I just I couldn't help. I tried to become a chemist, so I trained as a chemist, but biology kind of sucked me in. And I've been at Hui for more than 20 years. I was a graduate student, and then after a brief stint away, I was a postdoc, and now I work on mostly in toxicology and genomics. So interested in how animal evolution has shaped the genes and the enzymes that deal with the chemicals in their environment, and how the chemicals that we put in the water that we've produced, the humans have produced, interact with those systems to either make them, typically not to make them function not the way they're intended to. Helen, my co-PI on this is Helen Poynton, who is online. I'm sorry I can't make it bigger, or else I'll probably screw something up in Zoom, but Helen, go ahead and introduce yourself.
Hello, everyone. I'm happy to be here, even though it's virtual. My name's Helen Poynton. I'm a professor of ecotoxicology at UMass Boston. I've been teaching here for 15 years in the School for the Environment. And my science journey, I grew up with a very inquisitive father. He was into social studies and history, but he loved the science shows on TV, especially Nova. So we grew up watching Nova every week. And so I was really kind of immersed in that. But it was really when I was still pretty young, and my grandmother came to live with us because she was diagnosed with breast cancer. And she lived with us for about 2 years. And during that time, she got sicker and sicker and sicker. And at first, I wanted to become an oncologist and become a doctor to help people who had cancer. But I started, this is when I'm like 10 or 11, and I start reading about cancer and just became really, really interested in the cell biology that was involved in how cancer grows. And so that kind of led me towards this path of cell biology and molecular biology. At the same time, I became very interested in environmental issues. And as a teenager and a college student, I kind of became a environmental activist. Participated in a number of protests and stuff in the 1990s. Never did get arrested though. That was something that I've gotta I've gotta get on my part of my bucket list at some point. But I kind of had these two different competing interests that I was an environmentalist, but I also was really interested in molecular biology and just basic questions about how life is able to function at the molecular level. And fortunately, I went to the right graduate program. I thought I was going to graduate school. This was at UC Berkeley to study dietary effects and how diet leads to cancer. But I ended up in a lab that was starting a new project looking at how environmental toxins are affecting the genome of organisms. And in this case, it was a little crustacean called Daphnia. And to my surprise, there was this huge community of people who are all interested in the genome of Daphnia. And so I kind of got immersed into that little community, which was a really welcoming and nurturing environment to be a graduate student in. And that was the way that I was able to marry my environmental interests with my just basic intrinsic interest in cell biology. And so now a lot of, my research lab, we do a whole lot of different things. We look at a number of different model systems, mostly invertebrate systems. All of our questions kind of come back to the genome environment interactions, and in particular, how pollutants and chemical toxins are impacting that genome and how it's the genes and the genome itself evolves in the presence of these different chemical stressors.
I think with this proposal, this is what, the fifth proposal that we wrote together that finally got funded, something like that. So it takes a while, but Helen and I have been talking, working together for a long time. And then none of this would go without Alyssa.
Hi. So I am Alyssa Goncalves. I'm a fourth year PhD candidate now here at UMass Boston, and I work in Dr. Helen Poynton's lab alongside with Jed on this project. So thank you all for having us here, even though we're virtual. So how did I get here? I guess it's not the traditional path, but growing up, I was, always at the beach. It was a very, very big thing in my family to go to the beach all the time. And I would specifically go with my aunt all the time. Every weekend, we were at the beach. And every single time, I kid you not, she would make us stay. It's, 6 o'clock, 7 o'clock, the sun is setting. And we are the last people on the beach picking up everybody's trash. All the red Solo cups, the plastic bags, the chip bags, the sandwich bags. And I never really understood why. And I would be so frustrated as a kid because, you're at the beach all day, you're playing in the water, it's hot, I'm tired, I don't want to do this anymore. But a really, really valuable lesson came out of that. And it was, you can't just kind of turn a blind eye to all of these environmental issues, even if you're not the one doing them. So that's kind of where my passion for environmental science started. I guess it started with like marine science, focusing on the beach and things like that. I didn't know that it could be a career. I didn't know, people went to school to study these things, to study the trash that people leave on the beach, and what does that do to the ocean systems. I didn't really realize that until I was in high school, and I had a really, really good marine biology teacher. She was awesome, and I feel like she kind of flipped that switch to me and was like, you love going to the beach, this is something you love the marine environment, you think it's really cool, let's turn that into something. So I ended up going to my undergrad. I did my bachelor's at SUNY Maritime in New York, and I studied marine environmental science and marine biology. So I did some research there specifically on microplastics. And funny enough, before this, I was telling the story of how my undergraduate advisor, I keep her updated on the research that I do. And she's like, it's really funny, like you still work with poop water. And I was like, oh, okay. I mean, I guess, which is what we'll talk about is like these septic tanks. But when I was an undergrad, I used to work with, these combined sewer outflows in the Long Island Sound, and looking at enterococcus bacteria and what happens after storms, trying to answer those questions. So poop water is what I ended up doing. But overall, looking at different environmental stressors is, kind of what my dissertation focuses on. So that spans from like ocean acidification down to like this pollution stuff that we'll talk about today, as well as different community stressors and how those affect us as graduate students.
All right. So, Helen and Alyssa were actually just down in Woods Hole with me yesterday. We were beginning another experiment with the rib mussels, which I will talk about. But they're obviously, they had a meeting in Boston, and otherwise they'd be here again, although it's a bit of a drive. So I'm going to talk about two species of mussels, the blue mussel and the rib mussel. And they are both keystone coastal species. This is a AI generated slide, tweet slide. But just to sort of give a sense of one of the reasons that we are interested in them is not only because they create habitat and they create structure, but also they're really important for filtering water. So like the oysters, blue mussels have been a really important species for actually keeping what we think of keeping the water clear, keeping and cycling nutrients through the system. What has been observed is that there has been a steep population decrease of blue mussels, particularly in the Gulf of Maine, in mostly studied in coastal Maine. But I don't know if anybody here has observed it here I don't know about on Zoom land, but here has observed that there's also been a decrease around here, just sort of anecdotally. I haven't seen the numbers, but you did set up some questions. Mm-hmm. Just for my information, I can't remember what questions I gave you, but how many people live on Cape Cod? And this is a question actually for our coastal areas. So this is also a question for Zoom, since I don't know any people on Zoom. Mm-hmm. I presume everybody here lives around here. Mm-hmm. Yes. How many people actually live in Falmouth? Just for okay. The other question is if you know how your waste, your septic system waste is, your household waste is treated, whether it's a septic system or some other way, probably a sewer. Yeah. So most people are septic systems. So just and then the other one is, have you eaten a blue mussel? Yes. Has anybody eaten a ribbed mussel? No. One person. Okay, so I'll have to ask. No, okay, I'll have to ask. There wasn't
And we're getting responses online, okay. And we'll give it a little bit. We've got like 7 people who've completed it, but most people are on Cape Cod. Most people have septic system. It's split kind of if they've eaten a blue mussel, and no one has eaten a ribbed mussel.
Okay, so this is good to know. That's okay. No, it's fine. This. Is this going to keep on going? Can I just keep on going?
We can end the call. Yep.
Yeah. Just it was sort of for everybody's information. This is what's going on. It helps me. There's been a very steep population decrease. We don't know why, and it's probably a whole big bunch of combination of stressors, ranging from heat stress, which is something that Helen and I have been talking about. We've been put one proposal in. It could be parasite stress, which is something else we've been looking at. And Helen has a really, graduate student has a nice paper on that. But then more, but what we're talking about a little bit more here is actually skewed sex ratios. So this is something, I think Helen, you said it was started just noticing that there was, when they tried to sex ID a bunch of mussels, there was an awful lot of them that were female. And so they've actually done some more surveys, and there's definitely a up and down the coast, they tend to be skewed female. And that's sort of the background for why we're doing this research, is this observation of both the population decline and a skewing of the sex ratios. So the other part of this is thinking about what we're putting in the water. So some of these skewed sex ratios, I'm going to go back again, I'm sorry. If you see that they're kind of in more of the urban areas, and I think Scituate in particular had a high preponderance, and it was near their sewer outfall. So we started thinking about this in terms of the context of sewers versus septics, and point sources versus non-point sources of pollution going into the coastal waters. And this is a map of Cape Cod. This shows the sewer area of Cape Cod. Okay, there are approximately 250,000 people who live year-round on the Cape. It obviously swells greatly, probably at least threefold, or fourfold in some. And so clearly we're not doing much sewering. We have not done much sewering. And the proposed sewer area map looks very, very different. I'm not going to show that because it's proposed going, decades and decades into the future. So everybody says, most people are familiar with that they have septic systems. So whether or not they're familiar with the septic system, this is sort of more or less how it works. It is on-site sewage disposal, kind of a little bit of treatment, but mostly just disposal, sort of out of sight, out of mind. You have a tank, household waste, we call black water, poop and pee, flow into it. Gray water typically also flows into it. And you get a scum layer and a sludge layer, and then the liquid goes out into a septic field. This is a typical septic field. It's got sand underneath it, and it's embedded in gravel, and that's it. So it basically is kind of an out of sight, out of mind. You do get a bacterial mat that forms underneath, and there is actually loss of nitrogen, and there's some intense biological activity that goes on. But because you're putting typically a lot of liquid into this, it kind of washes down. And that's actually illustrated here, is that you're putting it into gravel and into sand, and then it's connected through the sand to the groundwater. This is particularly an acute issue on the Cape, and because we're a giant sandbar. There are other places in the United States, and I'll show you some maps, but Long Island, parts of Florida, they're basically giant sandbars. And so when it rains here, the water disappears very quickly into the ground. And the same thing happens with the effluent from the septic tanks. Percolation rates are typically, less than 2 minutes per inch. It just poof goes into the sand, and so it connects right down into the groundwater and then flows out into our ponds and into our coastal ponds. So these are conventional septic systems. This is recognized as a challenge, and we'll get to why. But one of the things that Helen and Alyssa talked about, I didn't really talk about as one of my particular motivations, but what are called the contaminants of environmental concern. They used to be called, or we used to call them, contaminants of emerging concern. I think it's pretty clear that they've emerged as things that you need to worry about. So we're calling them, to keep it as CECs, they're environmental concern. So all these things that we put into our body, put on our body, put into our household, that go down the drain, drugs, hair, personal care products, hair, shampoo, all this stuff contains a whole vast array of chemicals that are designed to do certain things. Some of them are designed to do certain very targeted things to us, medicine. They go out, they are rarely removed by a standard septic system, and they end up in the environment. So these range from plasticizers to drugs to hormones. So a pregnant woman can pee out 1,000-fold more estrogen than a non-pregnant woman, for example. This is a natural process, but hormones are extremely, extremely potent chemicals. Keep an eye on the time here. So many of these are what are called endocrine disruptors. So I mentioned estrogen. Estrogen works at extraordinarily low concentrations. It's a compound, a hormone that's evolved over millions of years to have a nice fit to the systems that it controls. We have made a whole number of chemicals that mimic, deliberately or not, the effects of estrogen. So things such as diethylsylvestrol, and you can see, I've put these structures up, you don't even know them, but you can note, when you look at them, the lower left of each one of them has this particular structure. It's called an aromatic ring, but and that is kind of the part that fits really nicely into the target. So tamoxifen is actually this is actually a metabolite tamoxifen, but tamoxifen is actually a breast cancer drug. It is designed explicitly to bind to the same receptors as estrogen to prevent the effects of extra estrogen on breast cancer. So this is the sort of thing that you might be taking and then peeing out into your septic system. And so this is a very specifically, very designed molecule to do something very, very potent. Nonylphenol is something that is very commonly found in many hair products. And it just is. It's one of those things that's just made to do a certain thing, and there's a lot of it. It's not as potent by any means, but it's because there's a lot of it, you end up having an effect from it. So what does these are all designed to have effects on human, or do they have anything to do with mussels, which is sort of the reason we're talking about this. We'll get to that in a second. What we want to do, and the reason that for our research is understand what the effects of these new septic systems might be. Do they have additional benefits besides what they're designed to do? And what they're designed to do is actually remove nitrogen which is a pollutant. It's basically a fertilizer for a lot of the things that grow nicely in our ponds. And these systems, these new systems, and I know it's clunky, but innovative alternative, I'm going to call them IA systems, if I slip up a little bit. They kind of manipulate the nitrogen cycle. And so what they do is you add air, and then you actually have a section where there's no air. And by going back and forth between those two parts of the systems, you actually can eventually turn the nitrogen from the urea that we excrete to nitrogen gas. And it's through intense microbial action that happens. So you really stimulate the microbes going back and forth between those two conditions, and they transform the nitrogen and eventually eliminate the nitrogen from the whole system. They're really lovely. This is an example of one that's actually not that is in piloting approval in Massachusetts, but is approved for use as an IA system in other states. But this gives you some idea of the scale and of the complicated technologies of these. this particular iteration of it. This is why these things, this equipment alone costs on the order of $25,000, not including installation. That's just the equipment. So that gives you some idea of this. The question that we've asked, and that we're not the only ones, but we're asking specifically in this context, is can this intense biological activity have other, in this case, beneficial consequences? So we're talking about microbes. They do all sorts of interesting things. There's more microbes in us than we have cells, for example. They do all sorts of things. Can this activity chew up these complicated molecules that we're putting into our septic systems? So we have a test subject. We actually have two different test subjects. I'm going to pass around a couple of shells, and you'll see why. They have this is a blue mussel. These they shouldn't be terribly stinky. This is that's numbered because that was a research subject who passed away. Right. This is a rib mussel, and I can pass this on the other side. This is rib mussel. That's also numbered because that was a research subject that didn't exist. It was full of sand. We thought. So these mussels actually are really interesting. So the blue mussels are ones that we eat. Many people have eaten them. There's a good aquaculture for them. They're the reason we did this. We've run some experiments, very similar experiments to the ones that we're running the rib mussels on the blue mussels. And I can show you the data from the blue mussels. The rib mussels are really interesting because they actually live, rather than being preferentially attached to rocks or in aquaculture in suspended systems. So they tend to be in, if you wish, less muddy circumstances. The rib mussels live on the edge of the marsh, and they form some of the physical basis of the marsh because they tend to be at the margins. If you go out, or when you go out, look around. You find them right around where the salt grass tends to sort of end. And they, just like the blue mussels, and like oysters, they're filter feeders. They are really important in helping the nitrogen cycle, the natural nitrogen cycle that forms and helps sustain the marshes. And so this is for the people online, I'm passing around rib mussels, but for the people online, this is they're actually can be quite strikingly pretty. They have these lovely colors in many cases. You can see on the right-hand side, you'll see they actually they do have kind of byssal threads to hold together. They also actually, you can see how they can form part of the salt marsh structure by holding some of the sediment. And I'm going to actually point with my mouse so people can see. So here they're holding some of the sediment. Many of these we cut away just because they're in the way when we're labeling them, but they also can be hosts for barnacles, for example. So they are really, really important on this part, this downs just above mean low water part of the salt marsh. Now, as sea level rises and the salt marshes become more inundated, these mussels maybe form a very, very important part of protecting the physical structure of the marsh. So if our septic effluent is doing the same thing to the rib mussels as we think it's doing to the blue mussels, we could be really challenging them in these coastal environments. And so and then here's the other part, if our septic systems, the new technology, the septic systems, which are designed for removing nitrogen, also remove other things, we may have this additional real co-benefit, unintended, but incredibly important co-benefit coming from this new technologies and the adoption of these new technologies. So here are some research subjects this was taken yesterday. We set up a big experiment. What we do is we take effluent from underneath septic systems. And this we can do because the Massachusetts Alternative Septic Test Facility test center actually has pipes collecting underneath full installations of septic systems. So they dig a big hole, they put in those pipes that I showed you, and then they put pipes underneath them to collect the effluent. And they can collect it from different depths. We then dilute it in artificial seawater and expose animals to that various diluted things. And we also have controlled test chemicals. Now, what we do ahead of time actually is we sex ID the individual animals. And the nice thing, the helpful thing about the blue mussel and the red mussel is that they are sexed, that they are a male and a female, and it's genetically sexed, and they have a certain gene program that is turned on and says, okay, this is a female, this is a male, and they're unlike, for example, scallops, which are hermaphrodites. So they have both male and female. So doing this experiment on something that's hermaphrodite, you wouldn't tell whether that's male or female, and who knows who's not. Sex determination in animals is all over the map. Like, it's not, there's XY, which people are probably familiar with, that's what humans are, but then there's ZW systems, and there's mitochondrial, doubly uniparental mitochondrial inheritance, and all sorts of complicated things that form the basis of PhDs, for example. But for this, it's very convenient that they're male and female, and we can actually extract their blood, their hemolymph, in this is called, and determine whether or not they're male and female before we do any experiments. Then we can expose them to chemicals. So here's a process. We were doing this yesterday. We actually numbered them. The blue mussel you saw, we numbered with the Dremel to carve the number in, but the rib mussels we discovered, or I should say Alyssa discovered, that if you Dremel them, you cut through the ribs and you can cut into the animal itself, and that's not good for them. So we actually use nail polish. We used quick drying nail polish, which it takes a little while, but so that's why they're beautiful colors. But you can see that we're actually dremeling. Alyssa, I think, and these are her hands, is dremeling a hole, and then we can stick a needle in and extract and analyze the DNA and the RNA, mostly the RNA in this case. Then we have had various volunteers. So then we do something called PCR, which is basically an amplification of the gene, so we can measure it. So we can measure whether or not they're male and female. And then we expose them. And this is an experiment. This is actually, this right hand, I think this is with oysters, similar sort of experiment. But then there's an example of the blue mussel where you can see the numbering. That was number 121. I can't tell you without looking at the data sheet whether it was a male and female. But we number them, and as long as we don't lose the data sheets, we're all good. Sorry, Alyssa. And then we can put them either if we're not exposing them, like controls can just go straight to seawater if we want. Typically what we're doing is we're putting them in jars with bubblers, and we expose them and change the water every couple of days, change the diluted septic effluent every couple of days for 28 days, which is a typical time that equilibration to chemicals occurs in certainly in blue mussels, and we're presuming in red mussels. So with the blue mussels, we can actually use chemicals and change the sex pattern of them. So we can expose male mussels, and this is on the left-hand side, you'll see there's the male genes are biased orange in this case on the controls, and then through exposure to estrogen, more of them become biased female. And then a drug, ketoconazole, which is an antifungal drug, is designed to interfere with the synthesis of certain. It's actually interfere with the synthesis of cholesterol, but it also interferes with a lot of synthesis of steroids, so other hormones. And so we have tried using it. I've been experimenting with it as a in terms of seeing if we can turn the females more towards male by inhibiting that biosynthesis of whatever hormone, sex hormone the mussels have. Which I have to put a side note in here just to confuse you a little bit. We're not, even though these animals we know respond to estrogens, we're pretty sure that they don't make estrogen the same estrogen that we have. So they respond to them, but the field in general, I'm saying, is pretty sure that they don't make very much of that particular exact hormone that we use and that fish use. They make something very similar. We just haven't tracked it down because somebody has to give us lots of money to do that. But yes, so it's been a chase now for on the order of 30 or 40 years to figure out exactly why they respond to estrogen, but don't seem to make the particular compound that I showed you the structure of earlier. Yeah. So these chemicals can also change what's called the gonadal index. I think this is there's one more graph. So, and that is the ratio of the weight of the gonads to the weight of the animal. So it means that since these animals, as you warm them up, as you feed them, they're starting to make eggs, they're starting to make sperm. If you treat them with these chemicals, you can suppress that particular biology. And so that would also tend to suppress their breeding success if they're just making less of that. They're less ripe, if you wish. So this is part of Alyssa's thesis. What we have actually done is compare the effluent from, this is what we did last year, and we're repeating this with the rib mussels. We compared the effects of effluent from a standard system that I showed you to that from a particular type of innovative alternative system. And you can see in this case, so this is a particular gene that is more female biased, and you can see actually what we're doing in this case is we're making the females more female, if you wish. And the males, in some cases, we're still trying to untangle this. We are also making the males more female, but we're doing it also in the controls. So we're trying to figure out what is happening exactly in our statistics are all screwed up. But in general, what we're seeing is that all of the animals, both male and female, became more female. We do know, because we sampled this water at the same time that the EPA sampled the water, we actually have chemical analyses from these waters. So we know, in fact, that, and I'm not going to show you the gory data because it's a big data sheet somewhere else. We know, in fact, that these systems, these IA systems, are actually better at removing many of the these chemical inputs than the standard Title V system. Both of them are okay at removing certain things, but the IA systems are really good at removing many things. We just don't know which of those chemicals, which of those hundreds of chemicals that the EPA analyzed is actually responsible for turning these mussels more female. So I'm going to kind of bring this to a close, because what we're doing in Falmouth right now and in Cape Cod in general is not only sewering, but we're also having to, we're under state regulations to reduce the amount of nitrogen going into our coastal embayments. And to do that, we're spending a lot of money on sewers, but we're also going to be spending a lot of money on these innovative alternative systems. And you can see where the green and the pale blue on the upper left in Massachusetts is where it is. These are determined, these are absolutely, you are required to reduce the amount of nitrogen going into your systems, into your coastal waters. Now, the pink is where it could be required to put in, to reduce the amount of nitrogen going into systems. It's not, the reason that it's not now required is because the studies to determine how much nitrogen needs to be removed have not yet been done. So all of this area of southeastern Massachusetts is very similar geology. And so there's a lot of, you can see there's a lot of coastal sand, basically. This is not unique to Massachusetts. So I'm particularly going to call out Suffolk County, New York, has a similar challenge. That's also a giant sandbar. And they have huge nitrogen problems. In some regards, they are in slightly different area, sort of time frame on this that we are. They're a little bit more advanced in some ways. Our technology development is more advanced than theirs, so we'll see how these work out. Florida also has a huge problem with nitrogen pollution, and those are areas in Florida where they actually have aerial like county-sized plans in place or in development to deal with their pollution, some of which is agricultural. They have the additional challenge that much of Florida is also limestone based, so water flows differently there than it does here. But they're also experimenting and also working on using these types of systems to reduce nitrogen. So scaling out now to more in the United States, this is the on-site sewage disposal, otherwise known as septic system prevalence in the United States. And you can see that there are many places where it's more than 40%. And in fact, like for example, in all of New England, about 50% of houses that have been built in the last 20 years are on septic systems. It is potentially a very large challenge because but at the same time, it's a large opportunity, a large demand for developing new types of technology that not only remove nitrogen, but also deal with a lot of the other things we'll put in the wall. So I think I've got about 15 minutes. I can answer questions from the room and from Zoom.
Yep.
So
Lori will help facilitate some of those online questions. So if you're online and you have a question, type it into the chat. And then does anyone here in this room have a question?
Yeah, I was wondering if gamete production is different in the mussels that have sort of become either more female or more male.
we haven't in these experiments measured gametes. Helen, I'm going to refer some of these to Helen.
Okay.
Because she has had grad students look at some of this. Helen, I think you're muted. I don't know whether that's you or is that.
Yeah, I'm here.
Did you hear that question about gamete production?
Yeah, so we've looked at, I mean, we haven't looked to see if the gametes are actually viable or not, but we have done histology, which is a way of looking at the tissue structure of the animals. And in the single chemical exposure, so when we expose them to EE2, that synthetic estrogen, we found that the male mussels had female gametes. So they were making oocytes. We don't know if those oocytes would be viable, like if they would actually grow into a little miniature mussel or not. But we did observe that they were present in the male mussels that were exposed to the EE2.
And that's actually when you're talking about playing around hormone systems, it's common in animals exposed to chemicals or even to sewage effluent. So for example, fish in England have been shown to be basically have what's called imposex, both male and female parts of the gametes. So whether or not they're viable, it's probably not actually.
Yeah. I have two quick ones. One, I'm wondering about the experimental design. Why do you need artificial seawater if these are originally mussels that are pulled out of the environment? That's one quick question. I mean, do they cleanse themselves so fast that you want to have nothing else in the culture media?
Well, we're mixing them with septic effluent, which has no salt in it. So in order to keep the salinity the same between all the treatments. What we actually do is make extra salty water and mix it with some fresh water.
Makes sense.
Right, so,
Other quick one,
Yeah.
that's makes perfect sense. Other quick one. When my wife and I were growing up as kids on North Shore of Long Island, and then we went to school in Rhode Island and then up to New Hampshire, back to Massachusetts now. It was phosphate down there. That was the big deal. And so I guess in Nassau, Suffolk County, we got all new detergents, phosphate's out of it, but did that suddenly fix the problem and now it's nitrogen? I mean, they're both fertilizers, right?
they are, because the response to phosphates was so strong and effective. All detergents now are phosphate free. Most fertilizers that are or should be sold in coastal impacted areas should be phosphate free. Phosphate is still a pollutant, but primarily in freshwater ponds. And so, because the nitrogen and phosphorus cycling is different in freshwater than it is in saltwater, phosphorus is still a concern. It is still a major concern. I haven't talked at all about the freshwater problems in Falmouth. For example, Oceanographic Pond is slated for what's called an alum treatment, which will precipitate the phosphate back into the sediment. Well, it still stays in the pond and it will still come back out of the alum at some point over a decade or so. But it's sort of a intermediate term treatment. It's less of an issue in marine systems because there's enough other things going on that the nitrogen is the primary pollutant. Yes.
My question also relates in part to your role with the Board of Health. It seems to me that the Commonwealth has given the towns of Cape Cod, including Falmouth, the responsibility to do a job of reducing nitrogen, but not the authority to carry out that role. At what point will your research into what are now called innovative and alternative systems result in them being standardized for use such that we can actually go from theory to practice in reducing nitrogen?
The state has not only required us, they actually have given us the authority to impose the requirement for IA systems. So for denitrifying systems. So there is one other member of the Board of Health in Falmouth here. But we have in fact required new construction to put in denitrifying systems. In certain areas.
In association with the Title V system as well. Right?
Yeah, it's part of a Title V system. So it's part of the equipment of a Title V system. Yeah. What the state hasn't done is given us money to do so.
Yes.
So that's the real thing is that if these systems, the other challenge is so a standard Title V system might cost the equipment and most of the installation, depending on your landscaping or whatever else, but we're talking about say it's 18 to 22,000 kind of ballpark, I think it's at the moment. The problem is that the additional equipment necessary to remove the nitrogen and remove the other pollutants costs on top of that costs another $22,000 roughly, depending on your system. So we are talking now about 40,000. 40 to 45,000. And then if you're in extreme circumstances because of where you live and there are boulders and whatever else, we've heard estimates as high as 80,000 for people getting quotes because of where they are. And so that's the real problem is that we have the authority. What we'd like to do is be able to make it cheaper.
Yeah. If the policy can't be implemented, it's not really a policy.
Right. Oh, it's definitely, no, we're definitely doing it.
Then that is fabulous. And
Right.
Thank you for helping me to understand.
Yes. Oh, absolutely. That's what we're here for. Yes, I think.
We have a question online. I'm gonna
yes.
bounce to them and then we'll come to you.
Right.
We have a question from Jeff. Do AI systems effectively remove estrogen mimics?
From the data we've seen, the answer is yes. That an IA system is pretty good. It very much depends on the technology as to how good, but many of these systems are and particularly the what are called the drip dispersal, the shallow systems are amazingly good at removing pharmaceuticals. And we don't know why. My suspicion, which has not been tested to my knowledge, is that it's because it's in the roots, it's fungi that live in that shallow, what's called the vadose, shallow root vadose system, because fungi are amazing at breaking down complicated molecules, like really superb. That's why we're not up to our eyeballs in lignin from the trees. So, yes.
What's the reasoning behind requiring to go Title V system in addition to the?
Well, the IA system is part of the Title V system, basically. So it's an extra component on top of or next to your standard Title V. So the Title V system is what has been typically been put in place since sometime in like the late 70s, early 80s, and then it was revised in the 90s as to exactly the design of those. These IA systems are additional components that go in kind of in line with your standards parts, the components. Because the standard parts of the components are really just a box and pipes into the sand. And the challenge is you really need to do other things. You need to make the microbial environment right in order to do this shifting of nitrogen forms back and forth to eventually get it to nitrogen gas.
So are you saying that the system won't work without the?
So if you don't have that, if for example you have this IA system and this additional box, this additional innovative, alternative, expensive part stops working, then your system reverts to basically just being a standard Title V, which protects us from the viruses and the diseases that would otherwise potentially come through, which is why these were originally put in. It's so that you didn't have poop going out on your lawn and then getting sick from the neighborhood getting sick from the fact that you're sick at home. That's the real reason that we did it. We put it underground because otherwise there's just raw sewage going out the ground. And so if your standard system, if your fancy expensive system stops working, it still will function and protect you and protect your neighbors from the bacteria and the pathogens that would otherwise make everybody sick. It's just that it won't protect the environment as well. That's the thing.
We have time for probably one more question.
All right, go ahead, Josh.
So the male to female ratio, you don't need nearly as many boys as girls. Do you know if what that ratio needs to be given however mussels procreate?
Well, okay, so these are broadcast spawners. A female can make as many as 100,000 eggs. So in fact, you still need a lot of males. If each female is making it's different. It's different than in invertebrates, for example, where there's a lot fewer eggs because there's a lot more energy going into the eggs. So you still need a fair amount of males. I don't know what the ratio is needed to keep the species going, but it's not like the 1 to 2, for example, that we need for fish in the lab. So
We have one more online question.
Yep.
From Eve, the implementation you mentioned for alternative systems for new construction, is that for the town of Falmouth or is that Cape-wide?
That is specifically for the town of Falmouth. Each town is coming to this in their own way. Other towns, I think Marion requires for new construction. It's sort of it's a town by town implementation at the moment. The original requirement from the state would be that if you don't have a watershed permit, a comprehensive watershed permit to have this great big plan to deal with your estuaries within a certain period of time, everybody in that particular nitrogen sensitive area was going to have to replace their systems with one of these IA systems, whether or not they were failing systems or they had new construction. They were all going to tear them all up. And so most towns have said, oh, we're going to come up with a good plan. And so Falmouth is in the process of coming up with 4 plans, because we have 14 affected estuaries rather than 1 or 2 like most towns do. So.
Thank you. Well, we are at the end of our time together. So thank you for the in-person audience. Thank you for our virtual audience. Before you get out of here, both virtually or in person, we do have a survey. If you're virtual, it's been shared in the chat. And if you're in person, I have some printed paper here. We just appreciate you filling out these surveys. It helps us see what is helpful in our programming. What do you like? What are you learning? So fill out a survey so that we can do more of the things that you want to see. And thank you, everyone. Our online audience, you can log off, and our in-person audience, you can get your stuff and make your way out, grab a cookie, have some coffee. But thank you.
And I particularly want to thank Helen and Alyssa for their evening too.
Thank you for having us.
Yeah, thank you.
You used a term I've never heard before, plasticizers, when you were talking about the organo compound.